Okay, good morning everybody. We'll start the class. So this is, let them come. This is Science and Tech PREvision class 1, isn't it? Okay. So I hope you have got some insight about the other subjects. So in this class, let me give some more insights on Science and Tech. Okay. And now since you people, except the online people, you people attended my PCM batch class. Now you will come to know what I taught in the class, what is that we are going to do in the PREvision. So it is going to be what? Mainly based on the questions, previous year questions. Now you will come to know whether what I taught in the class was relevant to your preparation or not. Yes or no? So because we are going to discuss the previous year questions, and then we will come to know, right? So whether really I was teaching relevant to UPSC pattern or not. Okay, first that you will realize. Okay, and the second one, and as I said earlier, so Science and Tech is not something like other subject, right? Where the other subject going to have what? The plain text. If you read it, or you read it, or Mr. X or Mr. Y read it, the plain text is going to be same for everybody. It is going to give what? Same meaning. But that is not the case with what? Science and Tech. It is going to have what? A lot of technical terms. Without knowing the technical terms, understanding science or the technology part going to be what? Difficult. Okay, difficult in the sense it is not what easily understandable if you don't know the technical terms. This subject involves more technical terms than I hope for the other subject. Yes or no? Are you understanding what I am saying? So that is why this subject seem to be little bit difficult. Okay, and don't worry, the people in the beginning itself is saying difficult, all those things. No need to worry. Just I'm saying the fact. Always it is there, right, with the students when compared to the other subject. For your UPSC preparation compared to the other subject, I should not say the names of the other subject. Okay, and when comparatively when we see the subject and you could see a lot of technical terms, and you could see that there is some sort of difficulty. Yes or no? And without understanding the technical terms, it is difficult to interpret a text. Okay, so in this class what I'm going to do is that I'm going to discuss on diseases first. As I said earlier, so in science and technology you just concentrate on mainly what? The diseases part and biotechnology part. Technology, when it comes to technology, 4 technologies are important. One is what? The biotechnology. In biotechnology alone, every year they are asking what? 2 questions, 2 to 3 questions they are asking, or at least 1 question will be there, or at least 2 questions will be there. So biotechnology you have to concentrate. Preliminary point of view only I am saying. So there are going to be what? Almost 12 question minimum is going to be there. Maximum 15 question being asked. Okay, in this area, science and technology alone, minimum 12, and some year they asked only 7 question only. Don't say, sir, some year they asked only 7 question, you are saying minimum 12, and an average I am talking. That is what I said in the video also, right? So minimum 12 questions, maximum 15 questions also been asked in this area. Okay, so in that, finding out or tracing out the 2 questions from biotechnology area is important. And similarly, every year there has been what? 1 question, 1 or 2 question from diseases alone. Okay, and for your kind information, last year they asked 4 questions from diseases part. 2021 they asked 4 questions from diseases part. Of course, not state question. 2 questions are state question. One is a combination of biotechnology and disease. Okay, that is mitochondrial disease, what we discussed. Okay, and of course the other question is what? The virus, bacteria based, just disease causing agent based questions, right? So like that, every year there has been 1 question, and again year 2, in 2018 they didn't ask a question from disease. In 2012, before 2013, 12, 11 and all, they didn't ask a question from disease. Okay, and by having that information, don't say some year they didn't ask. Yes, few years they didn't ask question from directly diseases, but the remaining year either they are asking 1 or 2 questions, and last year 4 questions. and you know why they increase number of questions from disease area due to the pandemic. Okay, and this year we hope so some questions also comes from the diseases area especially, and last year they asked one question from coronavirus, the question based on coronavirus also, but many of us don't know what is the question. When they asked in the exam, we was just sitting literally don't know what is the question. And this year we hope so some questions also comes from directly coronavirus, and 2020 we expected no question asked from virus. Okay, so like that there are at least one or two or four questions last year, like that minimum one question will be there in diseases part. So identifying one question from science and technology, what it makes sense. Okay, 2 marks, every marks count in that case. We are identifying area, definitely there is going to be one question, means we have to give what adequate importance for that area. Okay, so today just we will be discussing the questions and the concept based on diseases. Okay, in biology. Okay, we will be just discussing the topic or questions based on diseases. So this is what I was saying. See, in diseases alone, the year 2021 4 question been asked, 2021 19 to 2018 they didn't ask single question from diseases. 2017 1, 16 1, 15 1, 14 2, 13 2, and of course 12 and 11 there were no questions from diseases. Okay, now you see the recent trend, at least there is one question from diseases part, right? So in that case we have to give some adequate amount of importance to this topic, diseases. So just I'll give basic information, what is disease, how the disease being caused, what are the causing agents, that we will discuss. Then we will go to what the previous year questions from 2021 backwards we will go. You understand? So this is going to be, those who have attended my class earlier, this is just going to be a revision. These things we have discussed, and the people, those who are attending online, might be you know this one if you are a veteran, and if you are a newcomer, may not be. Definitely you are in the preparation, I hope so. And it is going to be what, just recall for you. So you recall what is the information about the diseases. So what is the disease? It is a condition of what? Condition of distributed functioning of the, condition of disturbed functioning, this is glaring, disturbed functioning of the body caused by what? Infections and defective diet, hereditary, environment. See, this is, you know, it is not a normal condition. We are affected by some sort of what, virus or bacteria or some external environment. Okay, that is our normal functioning will be what, affected by external factors or internal factors and whatever. Okay, that is called disease. And disease causing agents or causes of disease. So pathogens are biological agents. The disease can be caused by what? These agents, bacteria, virus or fungus, right? Helminths, whatever. And the disease can be caused by these agents, biological agents. And disease what? Also can be caused by nutrient agents, when there is a lack of vitamin deficiency, nutrition deficiency. Through that we will be getting what? Some sort of diseases, deficiency diseases. So vitamin deficiency diseases like beriberi, scurvy, night blindness. Then mineral deficiency diseases, anemia, carbohydrate deficiency, fat, protein deficiency diseases like marasmus, like that. So excess of food also what? Causes obesity. And then we have the chemical agents. In that we have endogenous agents, exogenous agents. Exogenous means like the pollutants, allergens, and endogenous means the excessive presence of uric acid within us, uric acid, and then antidiuretic hormone, which causes what? Diabetes insipidus, or insulin, reduced secretion of insulin. That is what we discussed, diabetes, right? So these are what? Chemical agents. Just I'm saying how the disease been caused, what are the causing agents. Okay, chemical agents we have, physical agents like heat, cold and radiation, sound, humidity, all these things might cause some sort of disease like stroke, and then impaired hearing. And then mechanical agents, so fractures, over mobility leads to what? Fractures, sprains or dislocation of the parts, and then injury, chronic friction. All these things are what? Caused by mechanical agents. And then genetic agents, due to variations in the genes, modification, mutations in the genes, will get what? Genetic related diseases. These are what? Disease causing agents. Okay, and what are the types of diseases we have? One is congenital diseases. That is what? The disease from your birth. This is from your birth, which are present from your birth. They are caused what? Might be based on the genetic abnormality. Might be caused what? Genetic abnormality, or due to metabolic disorders or malfunctioning of any organ. This is some of the example for what? The congenital diseases, like your hair lip, and then cleft palate. So like that, this is hair lip, cleft palate, and then what? Cleft foot. These are some of the congenital diseases from your birth, right? And then we have acquired diseases. Acquired diseases, those what? The diseases which occur after your birth, okay, due to some external factors. So the factors may be what? Into two. So we can, acquired diseases, we can categorize them into two. One is communicable diseases, another one is what? Non-communicable diseases. Communicable diseases, it can pass from person to person. Non-communicable diseases, no, there is no such possibility. Okay, so communicable diseases are caused by, what are the agents? The virus or bacteria or fungus. These are the causing agents of communicable diseases. And most of these are what? Carried by a vector. What is a vector in this disease case? Vector is a disease carrying agent. Disease carrying agent. For example, the malaria. The disease malaria is caused by what? A parasite. That parasite is what? Infects the mosquito. The mosquito infects you, the malaria. You understand? So here your mosquito is what? Vector. You understand? Plasmodium is the parasite which causes what? Malaria, and that affects the mosquito. Mosquito is what? In turn affects you. You understand? So there mosquito is a vector, and of course these are the small organisms which are what? Vectors for carrying various diseases like mosquitoes, sandflies, ticks. These are the common vectors which carries the disease. And non-communicable diseases, these are what? Just they are not going to what? Pass from one person to another person. They may occur due to the malfunctioning of some of the organ or organ system of the body. So it is again, in this there are various types. Non-communicable diseases, there are various types. One is what? Deficiency diseases. That is what we discussed. Deficiency diseases based on what? Deficiency of vitamins, minerals, carbohydrates, fats, right? So those diseases comes under this category, non-communicable diseases. And of course I've given the list. So there is one question based on the deficiency diseases. There we will be discussing about all these things. Okay, so just vitamin A deficiency, loss of vision. Vitamin B1 deficiency, beriberi. And vitamin C, scurvy. Vitamin D, scurvy, it is like your tooth related. I mean, if you are having disease, means tooth will just fall out, and this area will bulge. That is what scurvy. And vitamin D, rickets, not strong bone, soft bones. Okay, rickets. Then calcium, iodine, iron deficiency. So you know what is going to cause. Okay. Then cancer diseases. So cancers also what? Comes under the non-communicable diseases. So cancer is a disease caused when cells divide uncontrollably. That is how cancer forms, or that is the cancer. Then allergy, genetic diseases. Genetic diseases also what? Non-communicable diseases. So just since it is the genetic diseases, how from a cell we have a chromosome. Chromosome consists of what? DNA. DNA consists of what? Segments of gene. Okay. So there is going to be variation in the gene or mutation in the gene. Because of that, this has been caused. So genetic disorder is a disease caused when this is a disease in whole or in partly a change in DNA sequence. So normal DNA sequence, if there is what? Change in the DNA sequence, it is going to cause some diseases. Those diseases are called what? Genetic disorders or genetic diseases. So this is what I said, mutation in one gene. If there is change in one mutation in one gene, that is called what? Monogenic disorder. If there is mutation multiple genes, that is called what? Multifactorial inheritance disorders. And combination of gene mutation and environmental factors or what? By damage to the chromosomes. All these things happens. When these things happen, then we'll get what? Genetic disorders. When there is a damage to chromosomes and the combination of environment factors and the chromosome damage happening. So we'll get what? Genetic disorders. Just I have given main difference between the communicable and the non-communicable diseases. So in the communicable diseases category, we have what? This HIV, hepatitis A, B, C, measles, malaria, Ebola, pertussis, Nile virus, Zika virus, all these things what? The communicable diseases. Non-communicable diseases, maybe your cancers, chronic respiratory problems, diabetes. These are what? Non-communicable diseases, which means though disease can pass from one person to another person, these diseases what? It is not possible. And then what are the causes? Which causes the communicable diseases? Which causes the non-communicable diseases? The causes for communicable disease going to be what? Some of the microorganisms. It can be what? Virus or bacteria. This is what we discussed, right? And here non-communicable diseases majorly what? Your daily activities, daily routines, all these things. That is what I am saying. Non-communicable diseases are lifestyle oriented, majorly lifestyle oriented diseases. Okay. So high level of stress, increased incidence of diabetes, blood pressure, smoking, excessive consumption of alcohol, lack of exercise, lack of proper sleep, all these things, or sleep deprivation, all these things leads to what? Non-communicable diseases. And of course, when compared to communicable diseases, non-communicable disease impact is more, and that is what many of the people having non-communicable diseases when compared to the communicable diseases. This is more dangerous than that. Okay. Okay, fine. Okay, we'll go to the first question, 2021 question. The term ACE2, ACE2 is talked about the context of which one? And many of the questions in the science and technology area, it will be of this category only. Many of the questions in science and technology area, it will be of just A, B, C, D. You have to choose one. Okay, if you want to check it. Okay, so in that case, what? You should know what it is. And if you see the option, look at here. And genes introduced in the genetically modified plants. This is biotechnology area. Development of India's own satellite navigation system. This is what? Space technology. Radio collar for wildlife trafficking. Wildlife trafficking. This is related to what? Environment, something. And then spread of viral diseases. Disease area. See, 4 options in 4 different areas. Yes or no? Definitely one option is correct. In this case, what? You should know what is the meaning of that. Or else what? It is difficult to answer the question because options are not related here. If the options are related, at least you can come to know that elimination technique, you people say, right? Elimination technique I use there. So like that you can use when there is a relation what? Between these statements. Here all are what? Different areas. So that is how they are asking maximum number of questions. Okay. So in this case, this answer, so I have showed the answer. So answer is D, spread of viral diseases. How it is related to spread of viral diseases or virus diseases, that we will discuss. So that ACE2 stands for angiotensin converting enzyme 2 or ACE2 receptor. ACE2 receptor. See, this is the context. That is why there is a question on this ACE2. Where our coronavirus enters human cell through ACE2 receptors. It is entering, right? It need a binding mechanism. That binding mechanism is given by the receptor called ACE2 receptor. You have a protein, coronavirus outer protein, right? So the outer protein will go and insert in the receptor, like your key, how we are putting inside the lock. The same way, coronavirus will go and sit in the receptor. Are you understanding what I am saying? And in that year 2020, the question is in 2021. In 2020, there was an article. It is in the PIB, Press Information Bureau, written by some scientist. I didn't give the scientist name. Under the Ministry of Science and Technology. This is the article. It talks about what do we know and what do we need to know about novel coronavirus. It was the beginning. We understand? April 2020, almost we have completed only 3 to 4 months of the virus. That time people had what? Many ideas about the coronavirus. All become doctors. So this we have to eat, this we should not eat, this medicine you have to take, that medicine you have to take, this will affect this one, this. Yes or no? So many speculations, so many information, so many things were shared in WhatsApp and other social media. To curtail all those things, the government side, they have given clear instruction. Okay, what is the disease, how it infects, whom it infects, yes or no, what is the spreading rate, all those things clearly they said what you have to do as a precautionary measures. The article is about that one only. In that, there is one section, infection. He has written about the infection, how it infects. In that you see, the virus infects the epithelial cells, the skin cells, in the throat and lungs. Coronavirus. This virus binds to the ACE2 receptors. ACE2 receptors on human cells. So that virus going to what? Bind to the receptor. These are what? The just cellular doorway to what? The coronavirus. ACE2 receptors. These are like protein only. Okay. And which are what? Often found mostly in throats and lungs. These ACE2 receptor mostly found in throats and lungs. Virus on your skin lacking ACE2 expression will be harmless. The virus enters through by nasal passage, eyes and mouth. This we know. So the virus going to enter through these areas, and in these areas what is present? ACE2 receptor present. It is going to bind to the receptor. Which one? The virus. So it is what? Receiving agent. ACE2 receptor. Okay. There was article in that they have mentioned that this ACE2 receptor question been asked in 2021. You understand? Okay. So what is this? Just we will discuss what is this ACE2 receptor. We will get some more insight. Okay. See, this is a protein, as I said, on the surface of many cell types. This is the protein on the surface of many cell types, or you can say it is an enzyme generates small proteins. See, using a spike-like protein on its surface, what is the coronavirus binds to what? ACE2 receptor. This is what I said, like a key being inserted into a lock prior to entry and infection of the cells. Before entering and infecting the cell, it has to what? Bind. For that, this receptor being used. Okay. And this is what I said. ACE2 acts as a cellular doorway. Yes, it is like a door to the virus. And this is how it is. ACE2 receptor we have, coronavirus, and that protein we have. It is going to bind. Then it is going to what? Go inside, and it is going to affect the cell. You understand? And this is what that receptor is. This is how it is going to be. The question is, the question being asked like this. Now you come to know why in some areas I gone little bit deeper. You understand? Okay. So next question again you'll come to know why I have gone little bit deeper. Okay, fine. Okay. This I discussed. See here, your AS2 is present in epithelial medium, or where nose, mouth, lungs, in all this place it is present, and it is present abundantly in the lung area. Lung area it is present abundantly. And see here, AS2 helps modulate the many activities of the protein called angiotensin that increases. See here, that is one protein, angiotensin, that is going to what? Increase blood pressure and inflammation, increasing damage to blood vessels, and what? Various types of tissue injury. It is going to be caused. These are the what? Going to happen when there is what? Angiotensin, right, protein. And this is going to be modulated or controlled by whom? AS2 receptor. Okay. So this is what going to happen. AS2 converts this angiotensin 2 to other molecule that counteract the effects of angiotensin. Okay, just it is going to modulate and control the effects of angiotensin. Okay, so what is going to happen? The coronavirus comes in, it lacks what? It binds to the receptor. So when the receptor activity is now what? Controlled. When the receptor activity is controlled, it is not going to be active. In that case, then angiotensin going to do what? All the damaging works. And that is how the coronavirus affecting the lungs. You understand what I'm saying? So that is what it is. So when coronavirus binds to AS2 receptor, it prevents AS2 from performing its normal function. That is only what? Modulates the angiotensin 2. Now the angiotensin is what? Now free. It want to do what? The damaging work it will just keep doing. You understand? That is how this AS2 is important. That protein is important. If there is a decrease in AS2 activity, then AS2 cannot break down this angiotensin 2 protein, which means there is more of it to cause inflammation and damage to the body. You understand? So angiotensin 2 is controlled by what? Or modulated by AS2 receptor. When the coronavirus binds to the AS2 receptor, its activity what? Fully controlled. In that case, this activity, the damaging activity done by what? Angiotensin will be what? Improved or increased. That is how, that is the importance of AS2 receptor. You understand? Okay, fine. And I know, since we are discussing about the coronavirus, see, I didn't plan. Okay, the first question itself, what? Coronavirus based question. So let us just discuss again the information about coronavirus with respect to exam point of view. Just we are expecting this time like the question based on the various variant of the coronavirus. Okay, at least the what is that? Variation of concern, variation of interest, like that question based on that. So let us discuss that one. Okay, not exclusively about coronavirus, what is the symptom, how it spreads, whom it affects whom. No, not like that. So it is infection caused by what? SARS coronavirus 2 variation 2, and that virus also what? Reason for other diseases like SARS and MERS. SARS means what? Severe acute respiratory syndrome. MERS, Middle East respiratory syndrome, that we discussed. Middle East and SARS. Middle East respiratory syndrome, it reported in Saudi Arabia in 2012. Okay, coronavirus, all these things, this virus, I mean this disease, and this, which one? MERS and SARS, all are what? Similar only. Okay, all are what? Caused by the virus of coronavirus family only. Okay, and it has been reported in from 2012 onwards. This is what? SARS from 2003 in southern China, and it has been reported in 2002. And first infection 2002, where in Guangdong province of southern China. And of course, it has the own symptoms like coronavirus only. Other information are like what? Coronavirus only. But you know that this coronavirus is not the new virus of that family. Okay, already similar kind of what? Virus we had, and disease also we had, MERS and SARS. Oh. So it is what? The virus is RNA virus. So it is not a DNA. The genetic material is RNA based, not DNA based, and that you know. Okay, so you know, right, what is mutation, variant, stain? Could you tell me what is the difference between mutation, variant and stain? Multiply, yeah. Mutation means multiply. Oh, that is what you think. Mutation means multiply. Then what? Change in the genetic makeup. That is what? Mutation. You understand? The original virus will have its genetic makeup, RNA or DNA. Okay. So when it enters the body, it is copying, right? It is going to copy, multiply. When the multiplication or copying happens, the genetic material will change. Genetic material, all what? It will change. When there is a change in the genetic material of the virus, then we can say that what? Mutation happened. Because of the mutation, we get what? New variant. New variant. When we get a new variant, if there is a change in the behavior of the virus, if there is a change in the behavior of the virus, originally it will have what? Some behavior. Because of the variation in the genetic material, there is going to be what? Change. There might be change, there may not be what? Change in the behavior. When there is a change in the behavior of the virus to the new virus, then we call that as what? Strain. You understand? Change in the genetic material, mutation. Because change in the genetic material, we are going to get what? New variant. When the new variant we get, that might have what? Similar characteristic or behavior, or new characteristic, new behavior. When there is new behavior, then we call them as what? Strain. Okay. So know the difference between this mutation, variant, strain. This is what? Original virus, whether it can be RNA or DNA based. When there is a change in the RNA, mutation happened. We'll get what? Variant, change in RNA. And then what? New strain, change in RNA and virus behavior. Change in RNA and virus behavior, that is called what? New strain. So since we are going to discuss about the variants of coronavirus, you know how the name is given: alpha, lambda, and then delta virus, and then omicron virus. This we discussed already. Yes or no? So how? So WHO decided just to consider naming pattern by using what? Greek letters. If they use some other names, it might what? Affect the countries, many countries. So what? To avoid that, they have chosen the Greek letters to name the coronavirus. So we have alpha, beta, gamma, delta, zeta, all these things, lambda, kappa. So we didn't use what? Nu and Z, right? We didn't use Nu and Z. Straight away we moved on to the omicron. Okay, what is the reason? I said not Chinese president, many other surnames. Yes, that is correct. Many other surnames in China used to be what? Z. So it might affect the reputation of the country, or some cultural damage will happen. To avoid that, they didn't keep what? Z. And then what? New. So people will say what? New means they will say NEW. They will confuse with the new. So they didn't use these two. So straight away they have gone for next letter, omicron. Okay. Fine. That is what? Z happens to be popular surname in China. One person is having that name. Okay. Okay, and of course, they will confuse with the word new. Okay, then World Health WHO, World Health Organization classified each emerging variant of coronavirus as either variant of concern or variant of interest. Variant of concern or variant of interest. So, variant of concern, variant of interest, how they are different? Variant of concern. Okay. The variant for which there is evidence of an increase in transmissibility, higher rate of transmission, more severe disease, increase in hospitalization or deaths, and significant reduction in neutralization by antibiotics, and reduced effectiveness of treatments of vaccines. If these conditions are satisfied by a variant, then it becomes what? Variant of concern. Variant of concern, and there are what? 5 variants as of now in the WHO list of variant of concern: Omicron, Delta, Gamma variant, and then Beta Alpha. Alpha, Beta, Gamma, Delta, Omicron. You go in the ascending order. Alpha, Beta, Gamma, Delta, and Omicron is the what? Variant of concern. Then variant of interest. Almost all the things will be there. All are what? In severe. Okay. So less when transmissibility will be there. Transmission will be there, but less when compared to the variant of concern. Okay. Mainly what? It is going to be community transmission in this variant. Lead to what? Community transmission. All the things will be there, but everything will be what? In mild case. So in this category, variant of interest. Variant of interest, we have mu and lambda. Mu and lambda are in the variant of interest category, and variant of concern: alpha, beta, gamma, delta, omicron. Just remember. And I mean 2 days back, I come to know that some new viruses come, and what is that? Omega something. And I happened to see one article. It said that XE they said. Z, but I just studied that it is subunit of or a branch of Omicron only. A branch of Omicron or subunit of Omicron only. So okay, that also I have given. Okay, see, when variant of interest, the variant or the virus or the disease, right, which is in the variant of interest category, it can be what? Move to variant of concern if it is what? Satisfying these things. If it is known to spread more easily, cause more severe diseases, escape the body's immune response, and change clinical presentation, and decrease the effectiveness of known tools such as public health measures, diagnostic treatment, and vaccines. If it is simple, if the disease, the variant causes severe impact, then it will be moved from where to where? Variant of interest to variant of concern. In the beginning, everything what? Straight away they will put a variant in the variant of concern, or initially they will be what? In the variant of interest, then they will move to what? Variant of concern. Even delta is like that only. Initially it was in the variant of interest, then they moved to what? Because of the severity, they moved to what? Variant of concern. You understand? Okay. So this is what Z variant of coronavirus. So it is the Omicron variant, 2 prominent subvariants. BA1, that is the genetic name, BA1, BA2. This is what called recombinant Z variant, combination of both. It means it contains the mutation found in both the variant of Omicron. That is what Z. Your Omicron consists of 2 subprominent variants, BA1, BA2. 2 variants are there, right? So the new one, Z, variant of coronavirus, is a recombinant of both. There is a mutation, change in the what? Genetic material of the first subvariant, and also what? There is change in the genetic material of the second subvariant, Again what? They are fusing, combining together, they are giving what? New one. That is called what? Z. You understand? XZ. This is what new variant. And okay. So with that, the coronavirus based question comes to an end. Next we will move on to the hereditary disease. This question, I mean this area, mitochondrial disease, I discussed in the class. Okay, exclusively I discussed, and I asked a question in the test also. Last test only I asked. Yes, I asked a question in the last test also. So again we will discuss. Okay, this is 2021 question based on what? The mitochondrial disease. Passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy. This is right. The cure is therapy, mitochondrial replacement therapy biotechnology. You understand? Therapy either before or after in vitro fertilization of egg. Okay, that is the first statement. See, too technical. Yes or no? Too technical. That is how they are going. Sometimes UPSC becomes what? I don't want to say that word. Oh. A child inherits mitochondrial diseases entirely from mother and not from father. Okay. So these are the two statements, and of course you have to validate the statement. Which one is correct? Both are correct statements. Okay. I showed the answer. Both are correct statements. How? So here we'll have a confusion whether it is, they are just again when we read the question we'll have a confusion whether it entirely comes from the father. It should come only from the father only. Now why it is coming from mother, we'll have a confusion there. Okay, there we'll have a confusion where whether it is before fertilization or after fertilization. Before or after, we'll have only it should be before or it or either what, only it should be after fertilization. Like that, there we'll have a confusion. Here we'll have a father or mother confusion, right? I had the same confusion when I was. Okay, fine. The answer is both. Okay, so by using the mitochondrial replacement therapy, this mitochondrial diseases, right, can be prevented before or after in vitro fertilization of eggs, and this disease entirely what comes from the mother and not from the father. Just will give the supporting material or information to this. See, first you know what is mitochondria. I'll come back from the mitochondria. Mitochondria, it is what? One of the cell organelle. Yes or no? So it is a powerhouse of the cell. Why it is called powerhouse of the cell? The energy for many of the metabolism is generated by the mitochondria. Okay. So you know this is the mitochondria, and of course cell, the mitochondria. We are discussing about this one only. Okay. So the energy required for various chemical activities needed for life is released by mitochondria. That is why we call as what? Powerhouse of cell. And in the form of what? The energy released by mitochondria in the form of ATP, adenosine triphosphate. Adenosine triphosphate molecules. ATP is known as the energy currency of the cell. ATP is known as the energy currency of the cell. So, and don't think that only the nucleus is having the DNA, genetic material. Your mitochondria have their own DNA. Mitochondria have their own DNA and ribosome. Therefore, mitochondria are able to make some of their own proteins. The purpose of having what? Genetic material. They are the codes to generate what? Proteins. Right? So, mitochondria will generate its own protein. It is having their own DNA. It is having their own DNA. They can what? Produce own protein, and mitochondrial DNA makes up what? About 0.1% of the cell's total DNA. Okay. So about 99.9% is going to be in the nucleus. Remaining 0.1% from where? Mitochondria. Okay. And of course, it is not going to affect individual characteristics such as appearance or personality. Okay, it is going to cause some diseases. That diseases we can prevent. if you use the technology. You understand? Mitochondrial defects going to cause some diseases. By removing that, we can what? Prevent the disease going to the offsprings. You understand? That is it. And now we'll go to what is that mitochondrial disease. This is about mitochondria. What is that mitochondrial disease? It is a disease or disorder refers to what? Group of disorder that affect the mitochondria, which are tiny compartments that are present almost every cell of the body. Okay. The common factor among the mitochondrial disease is that the mitochondria, when it is affected, unable to completely burn food. When it is not burning food, it will not what? Generate the essential energy for the body. You understand? That is what it is. Symptoms might include what? When there is what? Damage in the mitochondria, will have poor growth, developmental delays, muscle weakness. These are some of the symptoms when there is what? Mitochondrial diseases. You understand? Okay. This is what the answer for that, second statement, the answer is here. While a child's DNA is mixture from both what? Mother and father, mitochondria are separate package of genetic that come solely from the mother. Solely from the mother. Why it is not from the father? The sperm contains very low number of mitochondria and mitochondrial gene. So sperm is having very low mitochondria or mitochondrial gene. So that is why the mitochondria is not going to pass from the father. It is going to solely come from whom? The mother. So in the offspring, the mitochondrial genes are inherited from the mother. Thus, a father with a mitochondrial gene defect cannot transmit the disease to his offspring. Only the mother with the mitochondrial disease will what? Transmit to the offspring, not the father. So this paragraph give the answer for the second statement, or second statement is true. So mitochondrial diseases are caused by what? Solely from the, caused by solely from the mother, not from the father. Okay. Next, the first statement they talked about what? Mitochondrial replacement therapy, right? That we have to validate. Mitochondrial replacement therapy, the new form of reproductive in vitro fertilization, which works on what? Principle of replacing a woman's abnormal mitochondrial DNA with the donor's healthy one. So abnormal and the defective mitochondrial DNA of the mother need to be removed, and it should be what? Replaced by the donor's healthy one. That is what this therapy is. Okay. So in that there are two techniques: Spindle's transfer and pronuclear transfer. And for your kind information, this is 2021 question. Pronuclear transfer technology. What is this technology? They asked in 2020. Previous year they asked. 2020. The question is on pronuclear transfer technology. 2021, one of the technology, this is the technology for what? For curing mitochondrial disease. That they asked in what? Mitochondrial disease itself they asked in the 2021. This technology asked in 2020. You understand? See, 2020 they asked one question in this area. 2021 same area they asked one question. Are you understanding? Okay. So there are this kind of, two techniques like that in this. So this is what pronuclear transfer technique. Just I'll save the diagram, shall I? We have the patient couple and abnormal mitochondria, and this is the donor normal mitochondria. This is the normal one. Then what? Injection of the sperm will happen. Right, so that means fertilization happens, and then what? We'll have the pronuclear zygote after the fertilization. Then what? We will remove which one? We remove this one. This is pronuclear. We remove this one. This is discarded, and this is what? The mother's one, pronuclear. That one will take and fuse it here. Okay, now we'll have this one. This is reconstituted zygote. This can be used to what, for further process. You understand? So this is what it is. See here, before, this is what, after fertilization. Fertilization happens, afterwards we are removing the DNA, mitochondria. You understand? See, healthy mitochondria are going to be here. Only that nucleus alone we are removing. You understanding? So your pro-nuclear transfer technique is what? Administered after fertilization. After fertilization. And whereas in the case of spindle transfer technique, this is before fertilization. How? So before sperm injection itself, you remove the what? The ear, the healthy one. Here the nucleus removed, and this is the patient one where we have the abnormal mitochondria. Okay, that abnormal mitochondria need not be carried. Here we have what? Normal mitochondria. Only that what? You take the nucleus and what? You remove it, you discard it, you insert here. Okay, then allow the sperm, do the fertilization, then use it for the further process. You understand? So this is what? After fertilization. Sorry, this is before fertilization. Yes or no? The technique before fertilization. That one is what? After fertilization. Okay. So in that case, this technology or this technique can be what? Used to prevent what? The mitochondrial disease before fertilization or after fertilization. Both are possible. You understand? So in that case, first statement is also correct statement. Okay. Okay, fine. That's it. And this is the concept for what? Three parent baby. Three parent baby. Three parent baby. This is what? Same. Your spindle nuclear transfer only. Spindle nuclear transfer means what? Before fertilization only, no? Before fertilization. And this is what it is. We have a faulty mitochondrial DNA from the parent's egg cell. What is that? So you don't want to send that faulty mitochondrial DNA, okay, of the mitochondrial DNA. Okay, that must be clear. Mitochondrial DNA, not chromosomal DNA. That must be clear here. And here, you have the healthy one, the nuclear DNA. What? Donor's egg cell we have. From this, what? We are going to remove that nuclear DNA and what? We are going to insert it here. Okay, same. We'll have the reconstruct, and this can be used to what? To create a baby. This is a baby formed by what? Involving three people: biological mother, and then what? The donor and the father sperm. So what? Three people. That is why this baby born out of this technique is called three parent baby. You understand? Okay, fine. And issues. Some of the issues, just each issue I can put as a statement, I can ask, no problem. You understand? What is the issue? The idea of baby having three biological parents, and argue that mitochondrial DNA goes somewhere to shaping the important characteristics of a person. But they are saying, scientists are not saying that no, there is no such possibility. It is just avoiding certain diseases. It is not going to what? Shape the character of a person or personality of a person. That is what the scientists claiming. Okay, one issue. Second issue, unnecessary. Some are arguing that the techniques are unnecessary. After all, it won't help those who have already been born with what? Mitochondrial disease. Those who are already born with mitochondrial diseases, this technique are what? Not going to be helpful, because it is happening in the embryonic stage itself. Yes or no? So it is not going to help. That one. Another one. See, this lasting genetic changes that will be passed down to through generation before we have a chance to find out if they are dangerous or not. So since we are making what? Changes in the genetic, we don't know what is going to happen in future. Okay, so there might be what? Some problem also because of this. Okay, changes. We are changing what? DNA. Some Of course, we are removing mitochondrial DNA only. Something might happen in the future. We don't have the evidence, okay, proper evidence as of now. So this is one of the reason, or this is one of the another issue. Okay, each issue can be what given as a three statement. I mean, one issue, second issue, third issue. Every issue what one statement. There will be one question. I don't know. I may ask, but UPSC I don't know. But know the issue. Okay, three parent baby. They'll give what is that three parent baby, and somehow the issue they can put as one statement. I don't know, because they asked that two question back to back. The next is only this one. They didn't ask straight away three parent. They didn't give that name three parent baby. Instead, they asked what the technology behind that. You understand? Okay, fine. This is the third question, and this is what I said. In the context of the recent advances in human reproductive technology, pro-nuclear transfer is used for 2020 question. Pro-nuclear transfer is used for, now look at here, as I said, right? Fertilization of egg egg in vitro by the donor sperm. Correct only, no? Fertilization of egg in vitro by donor sperm. No. It is not sperm. Fertilization happens for womb. Egg of the mother. Yes or no? Not from the, not by the donor sperm. It is donor mother egg. Yeah, you understand? Biological mother, donor mother. That's it. There is no place for father there. You understand? And then genetic modification of the sperm producing cell. Genetic modification? No. There is no, biotechnology. Genetic modification is not happening, right? In this, you are not genetically modifying anything. You are not changing any genetic material. Just we are removing the genetic material. That's it. Not making changes in the DNA. You understand? So that sentence is, second option is wrong. First option is also wrong. Development of stem cells into functional embryos. No. Development of stem cells. So it is not about stem cells. You know, right? Stem cells you can make as what? Embryos, and then what? You make the cells to function as per the requirement. That is not the technology here. Okay. Stem cell, the question based on stem cells also there. That we will discuss separately. Okay. So third option is not there. This is the fourth option is the valid and correct option. What it says, see, prevention of mitochondrial diseases in offspring. That is what your pro-nuclear transfer technology. Yes or no? So we can prevent the mitochondrial diseases which is passing through the offspring using this technology. Pro-nuclear technology is what? Before fertilization or after fertilization? After fertilization. Okay. So option D is the answer. Okay. I didn't give any further explanation for that. Now, the next one we are going to discuss about the virus. Disease caused by virus, because the next question is based on the virus only. Okay. So just we'll go fastly about the virus. Virus are what? The infectious agents that replicate only in the living cells of the organism. See, if it is separate means it cannot do anything when virus. You understand? It needs always a host to replicate, host to multiply. When it goes into the human, or when it goes to the animal, or when it goes to the planet, plants, not planet, plants, right? So it requires what? Host. Then only what? It will multiply and replicate. Otherwise, if it is virus means, if it is separate means it will not do anything. Just it will be just dormant. You understand? Okay, that's important. And of course, virus genetic material be what? It can be RNA or DNA. These are the some of the virus. Bacteriophage, which kills bacteria. Adenovirus, HIV. And this thing. Okay, this I have already said this thing. This is for the people, those who are attending online. So influenza virus caused by, just know the virus name, mysovirus. It affects which parts? Respiratory passage. So this is RNA virus. Influenza virus is RNA virus. Common cold. The question is based on a common cold only. Next question. Okay, here what virus I have given is, see, so there are a large amount of viruses which causes the common cold. One of the virus is what? Rhinovirus, which is RNA virus, and smallpox, variola virus, DNA virus. Smallpox is caused by variola virus, DNA virus, which affects, respiratory passage, then skin. You know what is smallpox. And chickenpox, varicella zoster virus, varicella zoster, and of course it is skin disease. Mumps, a paramyxovirus. Paramyxovirus, it is RNA virus. Again, it affects what? The salivary glands. So like that, that is the mumps. That is how it can be. Just swelling will happen. Okay. And then measles, again same virus which causes the mumps. Paramyxovirus, RNA virus. This is RNA virus, and again this is also related to skin only, skin disease. And then what? German measles, rubella. This is also, rubella virus. Virus is rubella virus. So this is also what? Skin disease only, rubella virus or German measles. Then polio virus, which is RNA virus. Polio. And then yellow fever, arthropod borne virus, RNA virus. AIDS, it is retrovirus, and that HIV is one of the what? A form of retrovirus. Okay, so general category is retrovirus. HIV is what? One of the subcategory. And then Ebola virus, and then Zika virus. So this is mosquito borne disease, Zika virus. Abnormal growth in the head, abnormal growth of head. That is what Zika virus. And look at the question. This is 2021 question only. Last year asked question. Adenoviruses have single standard DNA genomes, whereas retroviruses have double standard DNA genomes. They have gone deep. Instead of saying DNA, RNA, they have gone to what? Double standard or single standard. Okay, which is it correct? The second statement, common cold is sometimes caused by an adenovirus, whereas AIDS is caused by retrovirus. Second statement seems to be correct, because easily we can say retrovirus causes the AIDS, and adenovirus, there are many viruses, which even coronavirus also what causes the common cold. Yes or no? Cold. Like that, adenovirus also what causes common cold. Okay, so second statement seems to be correct. What about the first statement? Adenovirus have single standard DNA genomes, whereas retrovirus have double standard DNA genome. Single standard DNA. Single standard, it is called what? RNA. Okay, let it be. We can say it is single standard DNA also, no problem. You understand? Just now we discussed, what is that? Retrovirus, RNA virus. It is not the DNA virus. So your first statement is wrong. So in that case, what? That is why I have given that information about the virus. See here, like that they are asking based on RNA, DNA. Just they are again going deep, single standard, double standard, like that. Okay. So whenever I teach something deep, there is a meaning. You understand? Okay, fine. So B, only 2 is the correct statement, not this first one. Now just some basic difference. Adenovirus, these are medium sized, 90 to 100 nanometer virus, which contain what? Double standard DNA genome. Your adenovirus contains double standard DNA genome, reverse. Adenovirus contain double standard DNA, retrovirus contain single strand RNA. Okay, they are just reversed and given. Adenovirus can cause a wide range of illness such as what? Common cold, fever, sore throat, acute bronchitis, like that, pneumonia, pink eye, conjunctivitis. That is what the eye will accept that center part, other part, the white part will become what? Red. That is what it is. Pink eye. Like this. These are the some of the diseases caused by adenovirus, and also common cold is also there. Right? That is what the second statement for you. People with weakened immune system or existing respiratory or cardiac disease are at higher risk of developing severe illness from an adenovirus infection. So those who are having what? The immune system, weak immune system, they are what? They will be easily affected by this adenovirus. Right? Then retrovirus, a type of virus that has what? RNA instead of DNA as a genetic material. It is not DNA, it is RNA single strand. And it uses an enzyme called reverse transcriptase to become part of the host cell's DNA. And you see here the virus that causes AIDS, that is human immunodeficiency virus, HIV, is a type of retrovirus. This is what I said. It is a HIV is a type of retrovirus, and this is how it affects men. And it uses what I said, reverse transcriptase. What is it? Transcriptome means what? When DNA converts to RNA. Reverse means RNA to DNA. Reverse transcriptase. See, retrovirus we have. It is going to have what? RNA, genetic material, and that is the host cell. It comes in. When it comes in, it will use that reverse transcriptase. It will become what? DNA in the host. Using the reverse transcription technique, it is going to become what? Genetic material will be converted to DNA. Then it will attach to what? The nucleus or DNA material of the host cell. Okay. Then what? It is going to have the code. That code is what? Going to generate or replicate or whatever, copy the RNA. It is just going to what? Instruct to produce many RNA of the retrovirus. So it is what? Finally, we are going to have virus, retrovirus. You understand? So it is going to go as a RNA material, but inside the host it is going to become what? The DNA material using the reverse transcriptome. Okay. Then of course replication. And what about AIDS? I mean, the disease, it is a chronic disease. You know what is chronic disease and acute disease. Chronic disease lifelong. I mean, the duration is higher. Acute is what? Shorter disease. Okay, duration is shorter. And what is it? Of course, HIV interferes with the body's ability to fight infection and diseases by damaging what? Immune system. It is mainly going to damage what? Immune system. And these are some of the information with regard to HIV. ICMR, a study by ICMR, India is likely to miss target of eradicating AIDS by 2030. Eradicating AIDS by 2030. And we had promised that, or we had set a target in 2016 that India in the UN high level meeting on AIDS committed towards goal of ending the AIDS epidemic as a public health threat by 2030. Like this we committed, but now it is a study by ICMR says no, it is not going to be achieved. You understand? Next. And we have sustainable development goal. In that goal 3.3, the goal is what? By 2030, end the epidemics of AIDS, tuberculosis, malaria, neglected tropical diseases, and combat hepatitis, waterborne diseases, other what? Communicable diseases. That is the goal of 3.3, sustainable goal 3.3. Okay. And you know what is sustainable goal? There are 17 goals which are what? Need to be achieved by 2030. Who formed this sustainable goals? UN General Assembly. UN General Assembly. Okay. So fine. Next. There were an estimation says that 37.7 million people living with HIV at the end of 2030. 2020, not 2030. End of 2020. about 2/3 of whom are in African region. Okay. And in 2020 alone, 680,000 people died from HIV related causes and 1.5 million people acquired HIV. Okay. And WHO set the theme for 2021. This is the theme. Know the theme. End inequalities, end AIDS. Okay. Sometimes they are asking tag the theme. You understand? So end inequalities, end AIDS. That is the theme for the 2021 given by the WHO. Just for highlighting the growing inequalities in access to essential HIV services. Just to highlight that, they have kept this as the theme. Okay, when can you say that a person is progress to see? You know what is the main difference between AIDS and HIV? HIV is that virus which causes what? AIDS. AIDS straight away. If the virus infects you, you are not going to straight away going to get AIDS. There are stages, different stages, 3 stages. Third stage only what? AIDS. And if you are in the third stage, if you are having this diseases, means what is that happen? In here what? There is cell called CD4 cells. Its count should fall below 200 per cubic millimeter of blood. Okay. For a healthy person will have what? 500 to 1600 cells, CD4 cells. If he is infected with the HIV, you will have what? Below 200. Okay. Know the CD4 cell. Okay. Now, of course, there is no proper cure as of now. We have only treatment, that is retroviral therapy, just medication. Retroviral therapy, just giving medicines. Just we can save their lives, but we cannot totally cure the disease. Okay. What is that retroviral therapy will do? Reduce the amount of HIV in the body. Virus reduction will happen. Reduce the risk of HIV transmission. And what? Prevents HIV advancing to AIDS. Okay. And protects the immune system. And with respect to HIV, we have what? National AIDS Control Organization, NACO, India's voice against AIDS, which comes under which ministry? Ministry of Health and Family Welfare. Ministry of Health and Family Welfare, NACO. This is what leadership. It provides leadership to AIDS and HIV related control program. And almost we have 35 AIDS prevention and control societies. See, we have this program, National AIDS Control Program. It was launched in 1992 to control AIDS in India, HIV or AIDS, virus and the disease in India. Okay. So, Project Sunrise is there. What is the Project Sunrise as we discussed? Project Sunrise. This was launched in 2016. What is it? What? It was launched by the Union government to tackle the increasing HIV prevalence in the northeastern states. Northeastern states are more prone to the HIV disease. Just to tackle that or to contain that, they came up with this Project Sunrise. So mainly what northeastern state like Manipur, Nagaland, Mizoram accounts for the highest adult HIV prevalence in the country. Okay. So national average for prevalence of AIDS among the drug addicts is 7.14%, whereas in Manipur it is 12.9, Mizoram it is 12%. Okay. Some of the key facts, do you want me to say? That's it. The Project Sunrise, it is what? To tackle the HIV or AIDS in northeastern region. Okay. And it has the ambition of covering 1 lakh people. 1 lakh people. Then next we will move on to the next question. So bacteria, fungi, virus. See, if I need to discuss only the question, that is not it is not a test discussion. You understand? Test and the question discussion, that cannot happen. Okay. So question will be there as a base. With that, we will be just discussing what? All the comprehensive areas. You understand? Fine. Because why I explained briefly about, little bit elaborately about HIV means there is another question based on HIV. So that there I don't need to, I don't want to what explain. That is why I explained here. Okay, what is this? 2021 question only. But of course, this is what the medium or the causes of the many diseases. What are they? Bacteria, fungi, virus. The question is what? Which of the above can be cultured in artificial synthetic medium? Cultured, which of the following or which of the above can be grown artificially? Artificial medium. Which of the following or which among them can be grown artificially? All the above. Okay. When you are growing something artificially, you will have that medium only. Okay. They will have medium to grow the agar-agar. Mainly they will use what? Agar-agar as a medium, or the plate. Agar-agar plate they will use as a medium to grow bacteria or fungus. Okay. Bacteria, fungus have the ability to what? Multiply on its own. But whereas in the case of virus, if you just put the virus alone, it will not replicate, it will not grow. Virus always requires what? A medium, a host. The virus always requires a host to replicate. On its own, it will not replicate. You understand? So, virus in synthetic medium you cannot culture. No, no. The question is what? Which of the above can be cultured in artificial synthetic medium? Individually. Can you culture bacteria individually? Yes. Can you culture fungi? Yes. Can you culture virus alone? No. It always what? Need the support of the other cell or other tissue or organism. You understand? So, virus cannot be cultured artificially. So, in that case, what is the answer? Option A only. 1 and 2 only. Bacteria and fungus, not the virus. So, just artificial culture medium, also known as growth medium. Okay, in an environment used for the growth of different types of microorganisms. Just the most common growth medium for microorganisms are what? The nutrient broths or agar plates. This is what? Growth medium. This is how it is going to be. Okay, agar plate. Okay, the culture medium contains agarose and nutrients required for growth of the microorganism. Okay, this medium is first sterilized so that what? There is no contamination in it. So there is no other organisms grows. So first it need to be what? Sterilized. And bacteria, yeast, protozoans, fungi and algae are grown in the medium. I left what? Virus. Virus cannot grow. You understand? So bacteria, yeast, protozoans, fungi and algae are grown in the medium. A specialized culture medium containing, look at here, the extract of the plant and animal tissue is used for the growth of virus. Virus alone cannot grow on its own. We need what? Help of the, whether it is animal tissue or plant tissue. You understand? To grow in the growth medium or in the agar-agar plate, we need what? If you put just virus alone, it will not grow. Only what? We need the help of the animal or plant tissue. Okay. Okay. Just what is the for bacteria? For bacteria, nutrient agar and fungi, potato, dextrose agar. These are the growth medium. Okay, which are required. And virus cannot be cultured in artificial medium due to what? Lack of metabolic machinery of their own, and they depend totally on the host cell for replication. It is depending on the host cell. It always requires what? Host for the growth. On its own, it will not replicate. So, third, virus is not correct option. So, incorrect is what? Virus. So, option is what? A only. Okay, 1 and 2 only correct option. And what is that agar-agar? You know, it is gelatinous substance that is extracted from the seaweed and processed into, what is that agar agar? Did you see, you use in the food products, right? That is what agar agar. And given virus, bacteria, do you want the difference? No, right? You know what is the difference between virus and bacteria? Okay. And virus are not living organisms, these are living organisms, bacteria. Okay. And virus requires host cell to replicate, but it will replicate on its own. That is the main difference. Okay. And again I have given bacteria, fungi difference. And bacteria, they are mostly what, prokaryotes. That means single celled, nucleus membrane will be absent. Here nuclear membrane will be present. They are what, eukaryotes, multicellular or unicellular, it can be. Other things, leave it. Basic things, I hope you are knowing this, and if you want me to just tell all those things, I'll tell, no problem. Okay. Okay, now you see, which of the following are the reasons for the occurrence of multi-drug resistance in microbial pathogens in India? 2020 questions. Multi-drug resistance in microbial pathogens, whether it is India or any country. Okay, they are given specifically to India. What is it? Okay, multi-drug resistance in microbial pathogens. Genetic predisposition of some people. You know the meaning of genetic predisposition? This is acquired through the genetic makeup, genetic disease. Is there a relation between genetic disease and the multi-drug resistance in microbial pathogens? Genetic disease, that is caused by mutation of the gene, right? So nowhere they are related. First statement is wrong statement. Okay. Then taking incorrect doses of antibiotics to cure diseases. Multi-drug resistance. Using antibiotics in livestock farming. When you just farm this livestock, if you use the antibiotics, then it will be what? It will become as what? Multi-drug overuse. It is going to cause what? Multi-drug resistance. And what about multiple chronic diseases in some people? Multiple chronic diseases. See, the question is about the reasons for the occurrence of multi-drug resistance. Reasons for the occurrence of multi-drug resistance. What about the fourth statement? Multiple chronic disease in some people. Okay. The option is B only. That is what UPSC key gives. 2 and 3 only correct option. I'll tell you why. So now we'll start. What is antimicrobials? What are they? These are what? Medicines like your antibiotics, antivirals, antifungal. These are the medicines used to prevent what? And prevent and treat infections in humans, animal, plants. Antimicrobials. And then antimicrobial resistance means what? When bacteria, virus, fungi or parasites change over time and no longer respond to the medicines what you give, then what? We can say it has occurred the antimicrobial resistance. Okay, when it is obtaining antimicrobial resistant, what is going to happen? It is harder to treat, increasing the risk of disease spread, severe illness and death. Understand? So you are giving a treatment or medication for a disease, right? If it is not responding because of the change in the genetic makeup of the virus or bacteria, it is not responding, means then it has occurred what? Antimicrobial resistance. You understand? Okay, fine. Okay, when multi-drug resistant only, no, if it is what, not responding to multi-drugs, multi-medicines, then we say what? Multi-drug resistant what is that, organism or whatever, and that is called what? Superbugs. superbugs, they are what? Multi-drug resistant organisms or pathogens. Superbugs. So your antimicrobial resistant organisms, there can be virus or bacteria or whatever. They are found in what? People, animals, food, plants and other environment, in water, soil, air, in all the places we have what? This antimicrobial resistant organisms. Okay. They can spread from people to people, I mean person to person, or people to animal, or animal to people, and even from what? Food. Okay. Food of animal origin. See the main, they have given reason here. I showed this one. This is the reason given by WHO. Of course, over prescribing of antibiotics, causes of antibiotic resistance, over prescribing. Patients not finishing their treatment, they are not completing the course of treatment. Then what? Overuse of antibiotics in livestock and fish farming. Overuse of antibiotic in livestock and fish farming, and what? Poor infection control in hospital and clinics. Okay. And lack of hygiene and poor sanitation. Lack of new antibiotics being developed. So just to counteract. So these are the causes of what? Antibiotic resistance. Antibiotic resistance. And see that genetic predisposition means there is an increased chance that a person will develop a, this is based on the genetic makeup. Genetic makeup. Based on the genetic, okay, gene, there is a chance of what? Getting the genetic disorder, that is called what? Genetic predisposition. And of course the cancers, diabetes, obesity, these diseases can be what? Obtained because of the genetic makeup. And we are discussing about what? Which disease? That, I mean, multi-drug resistant organisms only, the causes only we are discussing. This is nowhere related to that. Okay, so first option are not the correct one. And the patients suffering from multiple chronic diseases will have what? Poor immunity. Okay, since they are given what? Fourth option, multiple chronic disease patients only, right? They will have poor immunity. Since they are having the poor immunity, they can what? They will be more prone to what? Multi-drug resistant diseases. You understand? And this is what I say. Whereas see, if the TB patients with HIV, they'll have what? More multi-drug resistant mechanism than compared to a TB patient without HIV. Are you understanding? So this is what it is. When the person is having what? More chronic diseases means his immune system will be what? Damaged. Because of the immune system damage, he is going to develop what? Multi-drug resistance disease. You understand? Okay. Then, and these are the some of the information about this multi-drug resistance. See here, there is global research on antimicrobial resistance report, which says 1.27 million people died in 2019 as a direct result of antimicrobial resistance. Okay, and WHO considers this as what? One of the top 10 threats to the global health. Okay, like AIDS and some other threats are there, and this is also one of the threats WHO considers. And your antimicrobial resistance is leading cause of death worldwide when compared to what? Which are higher than the AIDS and the malaria. Okay. And of course, the COVID-19 pandemic has also raised concern about what? Improper use of antimicrobials amongst COVID-19 patients. You know, in the beginning stage, what comedy happened? Everybody become doctor, everybody prescribed what? Some medicine. Yes or no? So, because of which antimicrobial resistance increased. So this is a study by ICMR from 10 hospitals showed that when COVID patients acquire drug resistant infection in hospitals, the mortality is almost 50 to 60%. This is a study by ICMR, and when COVID patients acquire drug resistant infections, the mortality, it is going to be what, 50 to 60%. Okay, fine. Then just we'll fastly discuss about hepatitis, because next question is the comparison between the hepatitis and the HIV. Okay, you know, hepatitis what? It is an inflammatory condition of the liver. It is a liver disease. First, you know, there are how many hepatitis? We have what? Acute hepatitis also, chronic also. In the chronic, it is showing what? No symptoms. Essentially, it shows no symptoms. Chronic. Okay. Whereas here acute inflammation of the liver that presents with the sickness. Okay. And you know, just I have given what is acute disease, chronic disease. I have said already. Acute is longer duration. I'm sorry. Chronic is longer duration. Acute is shorter duration. And what are the causes for hepatitis? It is caused by the hepatotrophic virus, liver directed virus. It includes what? A, B, C. There are five variations. A, B, C, D, E. Okay, five variations. And other virus also causes what? Hepatitis. And they are what? Varicella virus, which causes chickenpox. Of course, your coronavirus also what? Might cause the hepatitis. Okay. And other causes include your drug and alcohol use, fat buildup in the liver, and autoimmune process in which a person's body makes antibiotics. Right? These are the other reasons which lead to what? The hepatitis. See, types, as I said, there are 5 types: A, B, C, D, E. A is always acute and short-term disease, whereas B, C, D are most likely to become what? Chronic. And the E is usually acute but can be particularly dangerous in pregnant women. E. Okay. And just A, it is caused by hepatitis A virus, and here transmission, no, it is transmitted to others by contaminated food, water, and being in close contact with the infected person. Okay, it is transmitted to food. Transmission differs from ABCD. That is why I am explaining this. Okay, transmitted to by others through what? Food, water, and also being close contact with the infected person. And there is a vaccine. Hepatitis A vaccine is there. Hepatitis A vaccine is there. It transmits through contaminated food, water, and close contact with the affected person. B, so it is caused by hepatitis B virus, and it is what? Transmitted when sharing personal belongings, right, of an infected person. Also causes hepatitis B. B symptoms include abdominal pain, fatigue. Okay, these are the some of the, see, you imagine liver disease, that's it. Okay, hepatitis B vaccine is available. A, B vaccine is there. You understand? Okay. It contacted through flat tired wounds, contact with the blood, saliva, fluids of an infectious body. You should have the contact. Okay. And that one, it can pass through the food and contaminated food. Okay, water. And what about C? C, this is what infects at the time of birth for the child through the mother. It is passed on through using contaminated needles, syringes, or sharing other items with infected blood on them. Okay. So when it is infected person blood, through that also what? This disease passes, and it does not spread through food or water. Which one? The hepatitis C does not spread through food or water. Hepatitis A does, and there is no vaccine for hepatitis C. Hepatitis D, So it is again caused by virus, hepatitis D virus. It spreads from what? Infected blood or wound. Okay, that just remember this one, because the question is based on this one only, spreading, how it is happening. Sometimes it occur in the conjunction with the hepatitis B. Okay. Hepatitis E. Again, E is a waterborne disease caused by the hepatitis E virus. It circulates through food, water and contaminated blood. Okay. So A and E, food, water, contaminated blood. Okay. See, what is the complication of the hepatitis? Chronic liver disease and cirrhosis, chronic liver damage from a variety of causes leading to what? Scarring and liver failure, and liver cancer will happen. Okay. Chronic liver disease, long term, liver cancer will happen. Cirrhosis, it is a liver damage. Okay. So what is the complication? So bleeding disorders, buildup of fluid in your abdomen known as ascites, increased blood pressure in portal veins, kidney failure will happen because of this, and hepatoencephalopathy, which involve the fatigue, memory loss, diminished mental abilities, and then hepatocellular carcinoma, which is what? Form of liver cancer. All these things are the complication of hepatitis. Just this one, there are 5 types: A, B, C, D, E. We have vaccines for what? A, B, D, E. There is no vaccine for C, hepatitis C. And what? This is the affects hepatitis B and C, almost 400 million cases per year. Gradual death, an estimated 1.4 million people die worldwide from hepatitis every year, more than the HIV. And treatment, 90% of the hepatitis C patient can be healed within 3 to 6 months. Okay. See here, hepatitis A and E, this is what? Lack of food hygiene, contaminated water and substandard sanitary facilities. Through that it is spreading. And this one, BCD, blood, sperm and other bodily fluids. Okay. So basic information here. And this is another point. Hepatitis B and C are common liver infection among people with what? Living with HIV. These are the common infections or disease. People with people, those who are living with HIV will also what? More prone to have this hepatitis. You understand? B and C. So B and C together are the most common cause of deaths. Almost 3.24. They said, I said in the beginning, 3.4 million lives lost each year. And almost in 2016, 194 governments across the globe adopted WHO global strategy, which aims at what? Eliminating viral hepatitis by 2030. That is what 3.3, one of the goal, hepatitis elimination. And Indian scenario, so 40 million people are chronically infected with hepatitis B virus and 6 to 12 million people with hepatitis C virus. So we launched a national viral hepatitis program in 2018, and the hepatitis, okay, leave it. We have this information, right? I have already said, for finding hepatitis C virus, 3 people been awarded what? Nobel Prize for Medicine. Okay, in the year 2019. So already what? There is a Nobel Prize in 1976 for what? Finding the hepatitis B virus. Okay. Already for B and C, finding this virus, Nobel Prize been awarded in the medicine category. Fine. And this information you know, right? World Hepatitis Day is observed each year on 28th July to create awareness on viral hepatitis. But what you see here, 4 diseases, only 4 diseases that you must know. HIV AIDS 1st December, TB 24th March, malaria 25th April, hepatitis. The World Health Organization officially endorses disease specific global awareness days. Disease specific global awareness days. Okay, only 4: HIV AIDS, TB, malaria, hepatitis. Know the dates. Okay, now tell me which of the following statement is not correct. The question is not correct. Hepatitis B virus is transmitted much like HIV. Hepatitis B, unlike hepatitis C, does not have a vaccine. Hepatitis C only does not have a vaccine. Hepatitis A, B, C, D having what? Vaccine. The answer is what? B. That's it. Other 2 statements are correct statement. A is also correct statement. Okay. What is that? Option C says, globally the number of people infected with the hepatitis B and C virus are several times more than those infected with the HIV. Some of those infected with the hepatitis B and C virus do not show the symptoms for many years. So that is what I said in the beginning. Chronic. Okay. So for every point there is a reason. You understood? Okay, fine. So for this question, option B, that's it. Next question, consider the following statements. This is 2017 question. In tropical region, Zika virus disease is transmitted by the same mosquito that transmit dengue. Zika virus. Sexual transmission of Zika virus disease is possible. Both are correct statements. I should not show that answer. Okay. Fine. What is this about Zika virus? Zika virus caused by what? Aedes aegypti mosquito. These are what? Mosquito that spread dengue, chikungunya. That spread dengue, chikungunya, and also what? Zika virus. Which one? Malaria. That is also this one only, no? No. Zika can be passed through what? I mean, from sex also. If your partner is having that virus, pass through this, even if the infected person does not have a symptom at the time. Okay, at the time, if it is not having the symptom, then it can pass through this Zika virus. As I said, it is what? Passing through Aedes aegypti mosquito. Other disease, I mean chikungunya, dengue, yellow fever. Okay. Malaria. Okay. That will let you know. Then what? See, this Zika virus identified 1947 in Zika forest, Uganda. Symptoms, and just these are the symptoms. See, this is what when pregnant woman infected with the Zika virus may give what? Birth to babies with a severe brain damage or serious birth defects. That is called microcephaly. Microcephaly. This is what it is. Normal child, the child affected by Zika virus will have what? Abnormal head. Okay, that is what it is. There is no specific treatment or vaccine currently as of now for Zika virus. Okay, so accepted treatment, accepted vaccine is not there for Zika virus. Only prevention we have to do. Then next, which of the following statements is or correct? 2016 question. Virus can infect. Viruses can infect. Tell me bacteria, fungi, plants. Viruses can infect all. So it can be what? Just it need a host, that's it. If you are allowing the virus to any cell or any organism, it will affect it. Okay, whether it is a plant or animal or no problem, it can affect, infect. Okay, so bacteria, fungi, plants, all correct. right? Do you want me to give some basic idea? No, right? So it will affect all bacteria, archaea, fungi, plants, animals. It will affect all. Okay. See, based on they are affecting, right, based on the host only they are going to infect. Based on that, we can say plant virus affecting the plant, animal virus affecting the animal, and then fungal virus, bacterial virus. And these are the types of virus. This is how just I just put all these things, but time is not there. Okay, this is how the virus are going to be. Okay, so tobacco mosaic virus, it will be looking like this. Your adenovirus will be of this category only. Your coronavirus of this category. Your bacteriopages, it is going to be of this one. Okay, this is how virus will look like. Okay, H1N1 virus is sometimes mentioned in the news. What? With reference to which of the following diseases? AIDS, bird flu, dengue, swine flu. It is not bird flu. Okay, of course, AIDS, dengue you can remove. Swine flu, bird flu, swine flu is the answer option. Is a flu known as swine flu. H1N1 is the flu known as swine flu. The causes of swine flu is an influenza A virus designated as H1N1. Influenza, again there are 5 types, 5 or 4. Influenza A, B, C, D. Okay, 4 is there that I have given. Okay, the influenza. This is the this virus belongs to what? Large family of Orthomyxoviridae. Single standard segmented RNA genome. It is RNA virus. There are what? 4 types. Influenza A, which affects human, many animals. Influenza B circulates among humans, causes seasonal epidemics. Influenza C can infect both humans and pigs. And influenza D virus primarily affect the cattle and not known to infect or cause illness in the people, not human. So A, B, C will affect the human, not D. Okay, only the cattle. Just know. So you know this, right? Influenza A virus are classified into subtypes according to what? The combination of different virus surface proteins. They are what? I discussed this one already. Hemagglutinin and neuraminidase. Hemagglutinin H, neuraminidase N. They are what? Surface proteins. This is how it is going to be. Hemagglutinin and the neuraminidase. Based on that, they are going to what? Name the virus. Virus that has H7 protein and N9 protein is designated as what? Subtype H7 N9. You understand? Okay. So bird flu, avian influenza, caused by what? Also called avian influenza. This is what? Contagious and spread from bird to other birds and animals. Bird flu. Okay. Finished. It is caused by influenza type A virus, bird flu. Okay, which primarily affects what? Poultry birds such as chickens. Bird flu. And you know about the story, right? When the bird flu comes, the chicken rate will come down. Okay. Aquatic birds. Aquatic birds are primarily natural reservoir for most subtypes of influenza A virus. They are the virus pool. Okay, and depending on the origin, influenza virus can be what? Classified as avian influenza H5, N1, H9, N2, or swine influenza H1, N1, H3, N2, or other types, right? Influenza virus. H10N3. First human case of H10N3 bird flu was reported in eastern province of Jiangsu, China. First human case bird flu H10N3. Know that it is what? Type of influenza A virus. Okay, and the H5N1. This is in India, July 2021. First human death was recorded due to bird flu in India. First time. This was caused by H5N1 avian influenza virus. Okay, H5N1 avian influenza virus, first death. It is avian influenza is what? A type of, type A. Influenza A type virus. That usually what? Usually it affects poultry, including chickens, ducks, and it has what affected human, and death also happened. Okay, that is the important one. Okay, then of course this one, just 3 months after, see, we had this. Just 3 months after India declared itself free from avian influenza outbreak, bird flu, the year 2021 started with what? Unprecedented bird flu epidemic in 7 states. What are the 7 states? Kerala, Rajasthan, Madhya Pradesh, Himachal Pradesh, Haryana, Gujarat, Uttar Pradesh. Okay. December. Recently, first cases of bird flu have been confirmed in Kuttanad region of Kerala. Okay. And just tell me this question. Consider the following pairs. I was discussing vitamins and all, right? I didn't discuss unnecessarily about vitamins, minerals and all. Questions being asked, minerals, vitamins, based on questions being asked. Okay. See here, vitamin deficiency disease. Vitamin C, vitamin D, vitamin E. Which is correctly matched? Curvy rickets. 1 and 2 correctly matched. And vitamin E, night blindness. Vitamin A is night blindness, not vitamin E. Vitamin E, some fertility related things, I think. Okay. So option is what? 1 and 2 only correctly matched. We know night blindness is caused by what? Vitamin A deficiency, not E deficiency. Therefore, this is removed. Just I'll say what is the disease. Vitamin A, night blindness. B1, beriberi. That is what? Nerves and heart failure. And vitamin B2, riboflavinosis. Deficiency disease due to inadequate intake of riboflavin. And vitamin B3, pellagra. Severe vitamin B3 deficiency. Okay. Vitamin B5, paresthesia. What it is? Numbness or burning feeling that occurs most often in what? Extremities, arms, legs, all these things. Vitamin B6, anemia, right? Red blood cell. This is, and then vitamin B7, dermatitis, skin disease, and then vitamin B9, vitamin B12, megaloblastic anemia, a type of anemia. Vitamin C, scurvy, swelling of gums, that is what I said. And then vitamin D, rickets, condition that affects bone development in children. And also what? Osteomalacia. And then malacia. Okay. Vitamin E, less fertility. Vitamin E, they have given what? Less fertility. Deficiency causes less fertility. Vitamin K, non-clotting of blood. Vitamin K, non-clotting of blood. And shall we discuss or stop? Okay. With this, I'll stop. So, 2 more questions is there. In the next class, we will be discussing about the questions based on biotechnology area, and of course, that this is how I have to do. I don't know whether you like it or not. And questions, if I solve just a question, it will be what? Question solving session. No, it is the revision session. So, what is that information need to be given? That also I have to give. Okay. So 1.5 hours it is not sufficient for me, but anyway, I have more questions to discuss. I'll try to go with a little bit speed from the next class onwards. Okay. I hope I have given some information, and see, whatever we discuss in diseases part, the questions are based on those only. Are you understanding? And the SAR was taking diseases elaborately, biotechnology is taking elaborately, because questions been asked many times in those areas. What to do? You understand? So, and I don't know whether the online people understood properly. I hope you have understood. Is there any questions need to be asked? Do you have any questions? Any clarification need to be asked? Say yes or no, something, or text something. No, nobody responding. Okay then, here you people have any questions? No questions. Okay, now it seems to be what revision only, no, for you. That is how it should be. Okay, so I have given extra information also in the further questions. Okay, and what I have given, that also I will give, revise, and what has to be given as a input, okay, that also be given. Okay, so revision plus additional information, okay, along with the question. You understood? Okay, with this I wind up. Thank you.